All right, let's dive right into the incredibly fascinating, high-stakes world of the autonomic nervous system. In this explainer, we're looking closely at the cholinergic system. This is the biochemical communication network running everything from your heart rate right down to your individual muscle movements. But more importantly, we're going to explore how medical science, and unfortunately sometimes malicious actors, manipulate this very system.
I know, it sounds like the setup to some kind of dark riddle, right? But the answer actually unlocks our entire understanding of one of the body's most critical networks. You see, the venom of a black widow, the pills prescribed for cognitive decline, and the terrifying nerve gases developed for modern warfare, they all target the exact same biological mechanism, the cholinergic neuron, and its primary messenger, a neurotransmitter called acetylcholine.
To break all this down, this explainer is split into 5 quick parts: the acetylcholine life cycle, muscarinic versus nicotinic receptors, direct-acting agonists, reversible indirect agonists, and finally, irreversible agonists and toxicity. Let's get into it.
So, Part 1. The acetylcholine life cycle, or as I like to think of it, the biological factory.
Before we can understand how drugs or toxins manipulate the system, we really have to understand the baseline, right? We need to see how our neurons normally manufacture, deploy, and clean up acetylcholine. It's this highly regulated 6-step life cycle. Step 1 is synthesis in the cytosol. That's where choline and acetyl-CoA get combined. Step 2 is storage, where this newly minted acetylcholine is packaged safely into presynaptic vesicles. Then comes Step 3, the release. An electrical signal arrives, calcium channels pop open, and the vesicles just dump their contents into the synaptic gap. Step 4, the acetylcholine binds to its target receptors to deliver the message. Then, immediately, Step 5 is the cleanup. An enzyme called acetylcholinesterase swoops in and rapidly destroys the acetylcholine to stop the signal. And finally, Step 6, the leftover choline is recycled right back into the neuron to start the whole thing over again.
Now this is where it gets wild. Natural toxins already know how to exploit vulnerabilities in this exact factory. For instance, hemicholinum literally starves the factory by blocking the recycling of choline back in Step 1. Botulinum toxin acts as a massive roadblock. It completely blocks the release of the neurotransmitter in Step 3, which causes paralysis. But on the flip side, black widow spider venom attacks that very same step, but throws the gates wide open. It forces a massive, uncontrolled dumping of all your stored acetylcholine, throwing the nervous system into absolute overdrive. Moving on to Part 2, muscarinic versus nicotinic receptors, essentially the biological locks. So, the chemical key is out there, but it needs a receptor to fit into. And not all these locks work the same way. Muscarinic receptors are G-protein coupled. That means they trigger complex second messengers inside the cell, and they manage things like your heart rate, smooth muscle contraction, and glandular secretion. Nicotinic receptors, though, totally different vibe. They are direct, incredibly fast-acting, ligand-gated ion channels. When acetylcholine binds there, sodium just rushes in, causing rapid depolarization. You'll find these at the neuromuscular junctions, meaning they directly control your skeletal muscles and also at your autonomic ganglia.
All right, Part 3, direct-acting cholinergic agonists, basically, the master keys. Let's look at the pharmaceutical drugs designed to mimic acetylcholine and act as those master keys. By binding directly to the receptors, we can selectively target very specific medical issues. Say a patient has a non-obstructive atonic bladder, maybe right after childbirth. We can give them bethanechol. It has this strong muscarinic action that stimulates the bladder muscle, and it completely resists being destroyed by the body's cleanup enzymes. Then there's carbachol. It has both muscarinic and nicotinic action, making it a super potent tool used specifically in the eye to contract the pupil and treat glaucoma. And we can't forget pilocarpine. Because it's uncharged, it can easily penetrate the central nervous system. It is an absolute lifesaver for dropping intraocular pressure during glaucoma emergencies, and it's also incredibly effective at triggering saliva production for patients dealing with severe dry mouth.
Which brings us to Part 4, reversible indirect-acting agonists. I call these the factory managers.
Instead of directly mimicking acetylcholine, these drugs take a totally different approach. They temporarily stop the cleanup enzyme from doing its job, which causes your own natural acetylcholine to build up in the synapse. But how long does that actually last? Well, for a drug called edrophonium, it's just 10 to 20 minutes. It's an extremely short duration. Because it's cleared by the kidneys so fast, we don't use it for long-term treatment.
But that rapid action makes it the perfect, quick diagnostic tool for myasthenia gravis, which is an autoimmune disease where patients lack nicotinic receptors. By briefly flooding their system with their own acetylcholine, we look for a rapid spike in muscle strength, and boom, the diagnosis is confirmed.
It's really all about the physical structure with these indirect agonists that dictates exactly how we use them. For example, physostigmine can cross the blood-brain barrier right into the central nervous system. That makes it the ultimate go-to treatment for an overdose of an anticholinergic drug, like atropine. Neostigmine, however, is structurally polar. It cannot enter the brain, so it stays out in the periphery, making it perfect for managing the muscle weakness of myasthenia gravis without causing a bunch of central nervous system side effects. And then we have drugs like donepezil and rivastigmine. Patients with Alzheimer's disease actually have a deficiency of cholinergic neurons in the brain, so these drugs are designed specifically to enter the CNS and prevent the breakdown of whatever acetylcholine is left, which helps delay cognitive loss.
But as you might guess, there's always a catch. Manipulating this system is a super delicate balancing act. If you boost the cholinergic system too much, you trigger a massive cascade of systemic adverse effects. You are essentially forcing the body's rest and digest nervous system into extreme overdrive. We're talking profuse sweating, severe diarrhea, intense nausea, pinned pupils, urinary urgency, and really dangerous drops in blood pressure right alongside bronchial spasms. It is not pretty.
Finally, Part 5. Irreversible agonists and toxicity. The true saboteurs. This is where things get genuinely scary. We are talking about synthetic organophosphates. Take ecothiopate. It binds covalently and absolutely permanently to our cleanup enzymes. Once that bond forms, that enzyme is dead, finished. The only way the body can recover and start breaking down acetylcholine again is to genetically synthesize entirely new enzyme molecules from scratch. Now, while ecothiopate does have a very rare therapeutic use for glaucoma, its underlying chemical mechanism is completely identical to some of the most lethal toxins known to humanity.
Exposure sets off a terrifying biological ticking clock, a process toxicologists literally call aging. Here's how it plays out. In the seconds following exposure, the organophosphate binds to the enzyme. We then have a very brief rescue window, lasting maybe minutes to hours, where a drug called pralidoxime can theoretically jump in, break that bond, and save the enzyme. But then, aging hits. Within hours, the compound loses an alkyl group, and once that happens, the chemical bond is permanently locked in. After aging, reactivators like pralidoxime are completely useless. The patient is looking at days to weeks of brutal recovery, while their body scrambles to build new enzymes.
So why does this matter so much outside of a pharmacy? Because this exact irreversible mechanism is the foundation for common agricultural insecticides like parathion and malathion, which cause accidental and intentional poisonings all over the globe. Even more chilling, it's the exact mechanism of chemical terrorism agents and warfare nerve gases like sarin. These agents flood the entire body with extreme, unbroken cholinergic stimulation. Recognizing and treating this crisis is quite literally a matter of life and death.
If someone is in a cholinergic crisis from an organophosphate, saving them requires a rapid, intense multi-drug protocol. First up, pralidoxime, to try and detach the organophosphate and save the enzyme, assuming that aging hasn't happened yet. At the exact same time, you have to hit them with massive doses of atropine, that's a muscarinic blocker, to stop the patient's heart from slowing to a complete halt, and to keep their lungs from fatally filling up with fluids. Lastly, diazepam is given to penetrate the brain and shut down the violent, persistent convulsions caused by the sheer overload in the central nervous system.
So, looking at everything we've covered, I want to leave you with a thought. Right now, current cholinergic drugs act a bit like a shotgun. They hit receptors everywhere, giving us a mix of helpful treatments, but also massive side effects. But researchers are working right now to develop highly specific drugs that target only the M1 receptors in the brain, or maybe just the M3 receptors in the lungs. Will we one day cure conditions like Alzheimer's without triggering all those alarms in the rest of the autonomic nervous system? It's an incredible balancing act of biochemistry, and honestly, unlocking that perfect selectivity is the next great frontier in pharmacology. Thanks for joining me on this explainer, and I really hope it gave you a clear, structured look into the profound power of the cholinergic system.